This Bio 181 video is on chapter 11, which is on meiosis, the type of cellular division that results in genetic diversity and is associated with sexual reproduction. So meiosis only occurs in cells that are called germ cells. So that's a vocabulary word. Those cells are also referred to as sex cells. So those would be cells that are going to produce gametes. And gametes these are haploid daughter cells. So they have one set of chromosomes, and they're going to be either egg or sperm. So you can have a female gamete, that's egg, or male gamete is going to be the sperm, right? Meaning the male produces the sperm, the female produces the egg. During fertilization, these two cells, so you've got a big egg cell and a smaller sperm cell, okay? They're going to combine and form a single cell that is called the zygote. And the zygote will now have a ploidy level of 2n. So ploidy is the number of sets of chromosomes that an organism has or a cell has. So if it has one set, that's N, is haploid. If it has two sets, that's 2N, that's diploid, okay? So in this way, you regenerate the next generation. Once the zygote starts to divide, every subsequent cell is going to be through the process of mitosis. So then you're going to get two cells, and they're both going to be exact copies of each other, right? And then they're going to divide, and those cells are going to be exact copies of each other. So you're going to have a diploid organism. Okay. Sexual reproduction increases genetic diversity primarily through two major means, in addition to fertilization. One of them is called synapsis, which is associated with crossing over. Crossing over, that occurs in prophase 1 of meiosis. And then independent assortment. And so this is where it occurs in anaphase, basically, anaphase 1 and 2. When the chromosomes separate and go to the daughter cells, they go there randomly. So they could line up in the center on metaphase any which way, and then they're randomly distributed. So that's going to create a lot of genetic possibilities. And so let's look at synapsis in a little more detail, and then we'll look at the independent assortment. So in synapsis, you've got two chromosomes. One here I'm going to show is from the mom. So this would be the maternal chromosome. These are connected at the centromere, and they're identical exact copies. They've been replicated in S phase of interphase. So they've got the same sequence of genes in the same order, right? So this might be the gene for eye color, and this might be the gene for hair color, and this might be a gene that says how long your long bone is going to be, okay? And it's exactly the same in both copies, because these are sister chromatids, right? These are sister chromatids. And they were created when DNA made a copy of itself. So this original one here, if you followed it all the way back, if it came from the mom, it would be called a maternal chromosome, okay? And if you have the same thing from the dad, okay, so you have chromosome here, okay, it would have the same sequence of genes. So it's going to have eye color and whatever else, you know, does it make lactose enzyme? What's the hair color? How long is the long bone? So these are all going to be the same sequence of genes, okay, but they may code for different varieties of that gene. So say for eye color, say your mom is going to give you the allele for blue. So these would both be blue. And say your dad is going to give you the allele for green, just so I can see the colors here, right? And then for hair color, your mom might give you the allele for black hair, right? So the allele might be for black hair here. And then the dad might also have the allele for black hair. So that is possible, right? And then for long bone, maybe the mom is super tall. So like this is really long. So this is like very tall. It's going to grow super long. It's going to help you to be, if you want to be tall, a longer person, right? Whereas maybe the dad has a little bit shorter relative bones. And so for the dad, I'm just picking random colors here, but we'll just go with green, right? So this might be a shorter. Okay. So I'm just doing this to illustrate that we have different types of each gene. During synapsis, what happens is some of these are going to actually be exchanged between the sister chromatids that are closest together, okay? Not all of them are going to be exchanged, but usually there's probably at least 3 synaptic events when each of the pairs align. So afterwards, you're going to get on the left, this one is going to be identical, it's unchanged, okay, on the first chromosome. And similarly, on the second chromosome, the one on the right is going to be unchanged, okay? So the only change is going to occur in the one that is closest to the pairing in the center, right? So they've exchanged. So I'm just going to swap. So say this part moved over here. So now this sequence is from what was from the original dad. This sequence is going to move over here, okay? And they're going to swap. So they're swapping genes. So, and these recombinant genes now, right? You've got the allele for green eye color over here, okay? This one was blue. This one is, well, okay, this one is now blue here. And this one's green. And then I'm just going to do one more down here. So let's say for the tall and the long and the short, we have still the tall allele down here, but it's been swapped over here. And similarly for the short, okay? So now you've got these 2 new, what will become chromosomes once they're separated, with an entirely different combination than originally existed, right? The hair color on both of these like would still be black, right? Okay, so that's genetic recombination. Oh, and then for independent assortment, yeah, okay, I got 2 more slides. So for independent assortment, there's going to occur twice. So you've got the way that the chromosomes line up in metaphase is random. So the homologous chromosomes, you might get both of the, make it smaller, chromosomes from the maternal chromosomes on one side, or you might get them on opposite sides. So there's one example of how it might be oriented. And here's another example of how these may line up in metaphase 1. Okay, so it could line up like this. Okay. And then once they're separated, these are going to go to one side, these are going to go to the other side. And that's going to generate different possible combinations of chromosomes in the daughter cells, right? So this daughter cell over here is going to have these 2 small. These, I don't know why that's turning red. Okay, this daughter cell over here is going to have these 2 red ones, okay? Whereas in this case, we've got two other possibilities. Okay, two other possibilities. 1, 2. Okay. So we've got a big black X, there's a little one there, and we've got a little red X and a big one. Okay, so just from this different alignment, in this case during metaphase 1, how they line up on the left and the right, there's different possible cells, and this is going to occur both in metaphase 1, which is illustrated here, metaphase 1, okay? And it's also going to occur in metaphase 2. So in metaphase 2, how those sister chromatids separate to the daughter cells, that's also going to be independent assortment. Okay? So that increases genetic diversity. Then the last concept for this chapter is looking at non-disjunction. So non-disjunction is when we're separating the chromosomes and they don't separate equally. So typically you've got a chromosome. I'm just going to do the simplest case. Here's a chromosome in the center. And there's a big one. Let's say we got a small one here. Okay, small one here. Okay. The spindle fibers attach to the chromosomes, and they're gonna be anchored into these centrioles over here on the side. And during metaphase, the chromosomes line up, and then during anaphase, these are gonna get pulled towards the poles on opposite sides, and they're gonna separate down the middle. Typically, that's what we expect to happen. And we will get, okay, in the two daughter cells, small sister chromatids have separated and big. Okay, so that's what we would expect. So this is what would normally occur. However, in non-disjunction, what will happen is that, for non-disjunction, okay, we might get two daughter cells, and maybe one of them has the entire full sister chromatid still intact, like that, and one of them separated. So that would be one cell, the whole thing moved to one side. The other cell didn't get any at all. It still had one of, at least for the large chromosome. So in this case, we're having this cell probably will not be viable because it's missing an entire chromosome. So it will probably end up through apoptosis, not reproducing. This cell up here may reproduce, but it will have an extra part of the chromosome. This can also occur where we have extra chromosomes. So instead of just the sister chromatid failing to separate, we could have the entire homologous pair failing to separate and get an extra chromosome, which is what occurs in Down syndrome, where chromosome 21, you get an extra X chromosome, or you get an extra chromosome, not X chromosome, you get extra chromosome. It's called trisomy. And so that results in some changes in genetics. Right, that's the end for this video.